Hi, welcome to the podcast on genetic engineering. If you tried to access the podcast previously, it might not have had sound, so you might want to make sure you get the information down. So, what is genetic engineering? Genetic engineering is manipulating DNA for a particular purpose, and it's made possible by things called restriction enzymes. And most of the time, this particular purpose is for human purposes. And a lot of these pictures we actually saw in one of our videos where we have glow in the dark cats and we can grow ears on little mice and we can grow glow in the dark pigs and we can definitely engineer food to have certain vitamins and minerals and protein in it. And so we can do that by manipulating the DNA, and that's the maybe the joy of genetic engineering. But it also brings up some major issues and questions of just because we can do it, well, doesn't mean that we should. So looking at these things called restriction enzymes, which we worked at on in our DNA fingerprinting lab, and these are enzymes that cut DNA into pieces. Right? Different restriction enzymes cut a different sequences, just like we did in the lab. And so you're going to get just pieces of DNA so you can isolate certain genes that you want for a particular purpose. And when you combine that DNA piece with another organism's DNA, that's what we call recombinant DNA. Right? When pieces of the DNA from two or more different sources are combined to create something new. And this can be used for cloning or creating transgenic organisms or to create something in the cloning process that could even be, we could be even talking about something like insulin in the lab for diabetic patients. Cloning is a big process, though. It's a very big process that creates one or more genetically identical or copies of a gene or genome or even a full organism. And there's three types of cloning that we want to talk about: gene cloning, reproductive cloning, or therapeutic cloning. And the reproductive cloning is just cloning something new, like a cow or a cat or a dog. And the therapeutic cloning is more just cloning a tissue or spare part for medical purposes. We look at the gene cloning. All right, we're going to use restrictive enzymes. We cut that gene to isolate the gene that we want, and we take the gene out on, let's say, a bacterial cell to make room for the new gene, and then we combine the gene that we want and put it on the piece that we want to grow, and that becomes the recombinant DNA. And so every time this new cell over here reproduces, it's going to reproduce this particular gene. And this is typically how um, insulin is made in the lab. So again, every time that bacteria replicates, that gene is cloned. If we want the gene for insulin clone, that's sort of how we do it. All right, reproductive cloning. We want to reproduce something new, a new organism. And so we take there's three players in this. Well, two main players. Well, we have a donor cell that's taken from a sheep's udder, and we take the nucleus out of it. That's where the DNA is. We just need the nucleus. And then we take the egg cell out of an adult female sheep and remove the nucleus. Okay, so we have the egg of a female, then we have the nucleus from a donor cell, and we fuse them together. We put them together using electric shock, and now we have a fused egg cell, which is exactly what we want. That egg cell starts dividing and turns into an embryo, and we put that clump of cells back into the uterus of a foster mother, give it some hormones, and eventually it gives birth to a cloned lamb. This lamb is a clone of the donor cell taken from that original sheep, that nucleus. So that is how reproductive cloning goes. And this could, we don't have to be talking about sheep, but we could be talking about cows or dogs or monkeys. And of course, one of the videos that we watched, if we can do in monkeys, there's probably a certainty we can do this in humans. But again, I'll come back to this question of just because we can clone, does it really mean that we should clone humans? Therapeutic cloning is same concept here. We take an egg cell and we take the nucleus out. We take a body cell, we remove the nucleus from that. We put the body cell nucleus and the egg cell nucleus, or I'm sorry, the egg cell without the nucleus together. We fuse it, and then we insert that into, uh, or we flip a switch basically, give it some hormones, and that clone cell turns into an embryo. And so we take some of those, and we're not going to implant this into a female to reproduce into a human. We just want the stem cells. We harvest the stem cells from these embryonic stem cells, and that can turn into something like skin. That's how we sort of graft or regrow skin, right? We take a particular, we create embryonic stem cells and we take those cells and turn them into something that we need therapy-wise. Right, gene therapy is a similar concept, only we remove the bad genes and insert the gene that we want, usually a virus DNA, and we replace the damaged genes with healthy ones. Again, the key part is that we use virus DNA or virus nucleic acid for this particular process. It's just a way of treating the disease by replacing and removing these damaged genes. And does it work all the time? No, but it is a process. Gel electrophoresis. This is the technique used to separate the DNA molecules based on size and charge. We did this in the lab, or we did a simulation in the lab, where again the DNA sample is cut by restriction enzymes. We get different pieces of DNA, and then we load that into the gel, and we turn on the electricity, and those little bands, those pieces of DNA that we cut, are going to move at different rates. And so what we end up with is something that looks like this. All right, now everyone's DNA fingerprint is different. It is totally different because everyone's unique. And so the only one that might have a similar DNA fingerprint is an identical twin. But this is how electrophoresis works. Again, the DNA fingerprint is unique, and unique samples of DNA make unique banding patterns, and therefore no two people have the same DNA. And this is what we use in the crime scene labs, or even paternity cases, to figure out who belongs to who, or if you really were at the scene of a crime. Right, uses of DNA fingerprinting, forensics, of course. Right, we look at this here. We have dog 1, 2, 3, and 4. And it looks like dog 3 has the same band patterns as the culprit, so we could say that dog 3 can be found at the scene of the crime. Right, here we have another example, and we have the crime scene DNA, and we have the suspect DNA, and we look at suspect 2, 3 bands there, 2 bands there, 1 band here. Definitely was suspect 2. You can see the suspect 1 and 3 don't quite match up all the way. They might match up with some of the bands because we're all human, of course, and we are going to have similar traits, but it's not going to be an identical match, much like suspect 2. Here's a paternity case. Relatives have bands in common. So we have the mother here and the child share, and the alleged father share. So in this case, we would have that this is the father of the child. And if you look at result 2, where the alleged father has no matches with the child, so the result is the paternity is not established. Last thing again that we started with in the DNA replication, DNA structure podcast was the Human Genome Project. And this was complete. Whoops, get back to that. Completed in 2003, and the goal was to completely sequence all the human genes to identify locations of the various genes. And why would this research be important? And that's a really interesting question. Why would we need to do this? Just think of all the genetic diseases out there that we have no cures for. We'd like to have a cure for. And if we know where that gene exists, maybe we can find a cure for it. So lots of different reasons why we might want to know this, or if we want to even isolate specific genes and maybe even clone them in the future for medical purposes. Who knows? But this was a very recent study that was done and has many potential useful benefits. This concludes